I mentioned immunotherapy, so without further ado, let's kick off today's conversations. We're starting with one of the most exciting and promising areas in cancer research, as we've mentioned, immunotherapy, and we have some real rock stars joining us to tell us about their work and to hopefully make it understandable and not too wonky for those of us who are not scientists. Immunotherapy uses the patient's own immune system, as we well know, to fight cancer, either by boosting the body's natural defenses or providing the biological tools needed to attack the cancer cells. While some immunotherapies are used in combination with chemotherapy or radiation, it's within reach that they may one day replace both chemo and radiation altogether. Here to discuss their latest research in the field, you guys can come on in. I think this is the order we have you all in, but everybody probably knows all the distinguished panelists. Hi. I know them well. Dr. James Allison is here. Jim is chair of the Immunology Department at the University of Texas MD Anderson Cancer Center. Jim is right here. Dr. Carl June, the Richard Vague Memorial Professor of Immunology and Director of the Translational Research Program at the Abramson Cancer Center at the University of Pennsylvania. I don't know how you guys fit all this on your cards. Dr. Tak Mak, Senior Scientist at the Ontario Cancer Institute at Princess Margaret Hospital in Toronto, and Dr. Jedd Wolchok, who is Chief of Melanoma and Immunotherapy Service and the Lloyd J. Old Chair for Clinical Investigation at Memorial Sloan Kettering Cancer Center in New York City. And they're going to take a few moments to talk about their work. And Jim, let's start with you. Immunotherapy, as we know, has been around for over a century, but it really only took off recently, which is always surprising to me. Your discovery that cancer can put the brakes on the immune system and keep it from doing its job really revolutionized the field and led to a whole new area of immunotherapy that works to release those brakes, to take those brakes off. So what was your aha moment in language we can all understand? When did you say, wow, this is something, really huge development that is going to be so helpful in the war on cancer?
Well, the moment came when we were trying to understand the fundamental processes involved in regulating T cells, which are the army, the soldiers that go out and kill virus-infected cells, and potentially cancers, although up until that point they hadn't done it very well. And we found in studying the mechanisms, it was very complicated. Everybody before had concentrated on turning it on, but as you said, when we realized that there was a signal that turned it off to protect the immune system, to protect the body from being destroyed by the immune system, I had the idea that maybe that's why it wasn't very good at dealing with cancer cells, because the cancer got a head start, got very big, and if the brakes came on to stop the immune response before it had eliminated that last cancer cell, it wouldn't do it very effectively. And so I had the idea that if we just suspend the brakes temporarily until the tumor's gone, and then we can let it go and come back to normal, it might work. And our animal models were tremendously successful, and after several years we got into the clinic, and there were responders in the very first phase 1 trials. Soon became apparent that many kinds of cancers responded in melanoma, which was the first out of the gate. We know now through studies of thousands of patients that about a fifth of them, a little more than a fifth, are alive 10 years after a single treatment. And so this offered a lot of promise. While this work was going on, it was discovered that CTLA-4, this molecule that we've been working on, was the first of several checkpoints that were discovered. And the second one was one called PD-1. Anyway, we showed in my lab that you put them together and they're at least additive. And in the clinic, it was shown that you could treat, again, 50% or so of melanoma patients. So there's a lot of promise out there. There's optimism. We're actually saving lives. We're curing people. We know it can happen. It's not an issue of whether it can happen anymore. It's how good can we make it. And so the game has changed now, where what we're doing at many places, but especially in the immunotherapy platform at MD Anderson with Pam Sharma, we're doing studies of patients at baseline and then on therapy to see the changes that occur, the new markers that come up that tell us the new things that might be inhibiting, so we can develop rational ways of doing combinations that'll enable us to cure, hopefully, the majority of patients with many kinds of cancer. And again, I think the optimism's there, the promise is there. It's been shown we can do it. It's just a matter of putting our heads together and doing these detailed functional studies to to get there. But again, we're very optimistic about it because we're beginning to learn what the rules are. We're beginning to learn how to put this stuff together and again, save lives. And it's a great time to be in this field.
This may be a stupid question, which is often my specialty, but you talk about both approaches where the immune system has to be liberated to do its job, right? So the cancer is so wily and clever, it was able to. Part of the reason it could grow is it was suppressing the immune system, right? So without getting too technical, Jim, how were you able to get, what was involved in getting the immune system to be free to do its job?
Basically, it was just to take an antibody that was directed against this molecule that serves the brakes and block its function. So again, it couldn't work, the molecule that turned it off. We know now that there's some other ones that work slightly differently, but the approach is really the same, and most of it, that's just to come up with some way of disabling them temporarily.
And that's just through an IV treatment?
Yeah,
or?
it's basically an antibody that's infused for about an hour and a half. Typically, always it's just an outpatient. You go and get an infusion, you go home. There are side effects with these things, but they're manageable, and again, there's usually no hospitalization required. It's very simple treatment.
And is that used in concert with something that bolsters the immune system?
Yes, it's being used. We've discovered a couple of new molecules that actually give auxiliary on signals, stronger signals that are like superchargers, and we're beginning to target those as well. So we're learning. It's fascinating how to put all this together, but there's many targets we can work on, both to take the brakes off and to put the boosters on.
It's really exciting. And Dr. June, Carl, you were profiled in Cancer, the Emperor of All Maladies,
Yep.
A very moving part of the documentary that was produced and directed by filmmaker Ken Burns. And part of the series followed you and other doctors who were treating a young girl named Emily Whitehead. She was diagnosed at just 5 with acute lymphoblastic leukemia, and she was the first pediatric patient to undergo an experimental treatment. Her blood was taken out of her body, the white blood cells were removed and modified in the lab, and then returned to her body. Now Emily is 11 years old. She's been in remission for almost 4 years. What made you realize, Carl, that this experimental treatment might work?
Well, Katie, in leukemia, the one curative therapy over the years has been a bone marrow transplant. And that's done with someone else's immune system and with a lot of chemotherapy and radiation. Usually the other person's immune system is installed in the patient with leukemia. And it's a very toxic procedure. We saw yesterday Nick Wilcox, we saw his physician, Sergio Giual, who had done transplants with Robin Roberts. We saw some amazing results that can happen with transplants, but they have a lot of side effects. But that toxicity has been worthwhile to patients because it's been durable responses. So something Jim said is these patients seem to be cured. So the immune system, when it works, can make long-term responses, and it's not just a kind of weeks and months thing that we see with chemotherapy. So I saw that happen with someone else's bone marrow and in leukemia, and the field of bone marrow transplantation really started in the late 1980s. And so then a small group of people began working on making your own immune system be able to do this. And what we have worked out is something called CAR T-cells. And CAR stands for chimeric antigen receptor, and it comes after the Greek word from chimera, which was a beast made out of 2 different animals. And what we've done is make the immune system 2 different cells in one. And so our immune system, the T cells you heard about from Jim, we make a chimera with another cell called a B cell. And by doing that, we can get the best properties out of each cell. So we have had, since the mid-1990s, antibodies given to patients, such as rituximab. Those currently are given recursively, so every 3 weeks or so, and they're much less toxic than other therapies. And now with these modified CAR T cells, we only have to give them once. So the cells are given, and they're the first example of a living drug. So Emily, for instance, was infused in April of 2012, and she still has those chimeric T cells in her, and the same thing with Nook that we saw yesterday. So they're a living drug. You can give them once. They take advantage then of a convergence, as you mentioned, of different technologies that we never had before. So we now have a toolbox to make cells better than actually our own immune system is. So we can make in each person an Olympic immune system, if you will. So some people get over infections sooner than others. Some people reject their own cancers. But now what we can do with these kinds of antibodies and modified cells is make everyone have Olympic level immune system. So that's the goal. We have a number of new technologies, genetic engineering. You may have heard of things such as CRISPR, ways to modify cells to make them better than what we have. So it's a really exciting time.
It is exciting. And is it easier in children? I'm just curious. You mentioned Revlimid. My mom had lymphoma and ultimately passed away at 91. But she, had Revlimid and Rituxan, which I guess, would that be considered an immunotherapy?
Yep.
And I guess the question is it easier to boost the immune system in young people where their cells are a little, how shall we say, younger and healthier and just seem to be, in better shape in some ways?
So
less worn.
What your mom had was what's called targeted therapies. I mean, we're moving towards things that you mentioned, unfortunately, what your husband had to go through, which hadn't changed in 50 years, which is really a shame. So now we're moving towards personalized therapies that are targeted. So each tumor will be tested. And what your mother got is called, chemoimmunotherapy. But it was targeted in a way that, they didn't used to do in the past. And now it turns out that our immune systems are much more potent at younger ages. I mean, I think people have seen, vaccines work better. And as you get older, vaccines, you have to really boost them more to make them work. So part of what this field will do, I think, is try to regenerate immune systems back to what they were in our youth. And in fact, we've found now that children like Emily need less of these cells than the older patients that we treat. So that's, in fact, true. The immune system is more potent when we're younger.
I want to move on in a second, but before I do, Dr. G, I want to ask you about the serendipitous realization that you had when treating Emily Whitehead, that another drug might actually be very efficacious in her treatment, because that was such a dramatic moment in the Emperor of All Maladies, and it was pretty extraordinary. Can you explain briefly?
Yeah, I think it was a moment of desperation. So Emily had fevers of 106 degrees, which is 41 degrees Fahrenheit, for about 3 days. So she had, the first child that we had treated, a very violent reaction. And we were unable to stop it. And it turns out my daughter, who was here, had arthritis for many years. And a drug had just been approved for pediatric arthritis that turns off the immune system, but without being in a way like a sledgehammer is, like when we give steroids to really shut the immune system off. So in Emily's case, we tried this drug that's used normally for arthritis. It's called tocilizumab. And within hours, her fevers went away and she woke up. And the leukemia was gone. So we now use that routinely as a therapy.
Isn't that crazy?
Yeah.
Jedd, in fact, I want to talk to you about your specific research in a moment, Jedd, but talk about one of the challenges for immunotherapy is that the immune system gets almost hypercharged, and the impact of that hypercharged immune system can present its own set of problems, right? In fact, there's a name for that.
Yeah, that's right, Katie. We call it outside of this field autoimmunity or autoimmune disease, and the side effects that we've associated with these new medicines have been termed immune-related adverse events, and they basically represent an itis of almost every part of your body.
Isn't it called a storm?
So when it happened in Emily's case, it was traced to a storm of immune hormones called cytokines. And so that's really at the root of the approach that Carl's team used in trying to tame these side effects. So the immune system, like other organ systems in the body, has its own set of signals and hormones that it uses to tone itself up and tone itself down. And these cytokines can be very powerful forces in that.
Have you had those issues when treating your patients?
So we have noticed a series of side effects in treating people with some of the medicines that Jim had mentioned, these antibodies that block these critical brake pathways that the immune system uses to control itself. And in addition to tumors regressing, which is of course what we're aiming for, we've seen side effects, and they include rashes, which we can usually take care of with steroid creams, but they can often be more serious. Inflammation of the intestines, colitis, inflammation of the liver, or inflammation of the lungs. And this requires a bit more of a powerful systemic approach, where we give immune-suppressing medicines, either by vein or by mouth, which is sort of ironic, because
Right.
you would say, well, how could you possibly do that and expect these medicines to affect the cancer? And through a mechanism that, to be honest with you, we're still trying to understand, there is a disengagement or uncoupling of the toxicities from the benefit. And so we're able to safely treat the side effects in a very successful way without interfering with the anti-cancer activity.
It's a very delicate balance,
It is a
isn't it, Jedd?
balance, yeah.
And it was very inspiring, because Jedd has had incredible success with some melanoma patients, and we recently had an event, and it was extremely inspiring to see Jedd stand next to his patient, a woman named Karen Taphorn. Her melanoma had spread to her lungs, and she had 24 metastatic tumors. And she was treated with a combination of immunotherapy drugs, and now, incredibly, she is cancer-free. Were you incredulous when you saw, Jedd, how effective immunotherapy was for Karen Taphorn?
I've never been incredulous about immunotherapy, Katie. I think that's
Were you delighted?
I was delighted. I think that anyone who remained in this field for as long as all of us did could never be incredulous. It would have been too easy to give up. And I think it was our commitment to seeing the science through to true medical care that made the field what it is. There was a lot of effort in basic science laboratories to understand the immune system, and we knew that there was going to be a way forward. It just took a while and a lot of commitment. I think that the number of people who benefit with melanoma is very high. The way in which they benefit, which can sometimes be very rapid and dramatic, is a bit surprising. But of course, we're really glad to see that.
Did she have a lot of side effects?
So Karen actually had no side effects. In fact, when she finished treatment a few years ago, and I think that's an important part, and it goes back to what Carl and Jim both said, that there's a durability to this treatment, that you don't need to keep giving it. It's a dynamic treatment, whether you're stimulating the immune system with an antibody or giving cells, that this is triggering living cells in your body to remember the cancer and to keep reacting to it if it should pop up again. So I think that it's fantastic that when Karen continued to do well after she completed treatment, but she was wondering whether all of a sudden she would feel different than when she stopped treatment, that she had suppressed the idea that she was feeling unwell, but no, she feels just the same now.
But isn't it too early to say that you don't need a tune-up, or that people might need a little boost of immunotherapy down the road just to ensure that it keeps working well?
It may be a different answer for each person, and so we're following all these patients really carefully over time. As Jim said, there are some people who were treated 9 or 10 years ago with just a few infusions of these medicines, and they continue to do well. There are other patients who have had a good effect and then have had a recurrence of the cancer, and we've treated them again, and the cancer has once again gone into remission.
But if I could just add one thing, there's a woman who was in the phase 1 trial in 2001. We got a single injection of NSC-4. She's still doing fine without subsequent treatment 15 years later. She's probably the oldest person out.
But I'm imagining monitoring her very carefully, right, to make sure that her immune system remains supercharged, right? Tac Mac, my friend Tac Mac. I'm sorry, I don't mean to ignore you, Tac. Tac is another incredibly gifted scientist. I mean, we are actually in the presence of genius right now, but Tac is known for discovering the holy grail of immunology, the T-cell receptor, and this was back in 1984. And Tac, can you explain for us what a T-cell receptor is and how you were able to discover it?
Well, I think the best analogy in this context is that the T-cell receptor is the key to a particular car. So your key will only fit your car, and the reason why is that the viruses and the bacteria have to be recognized by different cars. So there are about 10 billion of those different kinds in your body, and they go up and down your body searching for viruses and bacteria and parasites to kill. Now, in this context today, we see that the analogy extends to after you turn on the car, you push the accelerator, and that is called CD28, and the car goes. And you kill, in this case, instead of the viruses, the cancer cell. And what Jim and Jedd have basically done is to say, well, what if I cover the brake? Then the car will just keep going and kill more and more killer T cells. And so I would then say the T-cell receptor is the key to a different car, and there are 10 billion of them, recognizing different bad guys in you. And the CTLA-4 PD-1 are the brakes, and the accelerators are other ones like CD20, OX40, et cetera, et cetera. So what we have seen the last few years, and Jim on my left is the pioneer, was to recognize using those brakes. And pharmaceutical companies and academic individuals are now playing with the many, many different brakes, PD-1, LAG-3, TIM-3, and playing with the many, many different accelerators. So what I, as a young man, I don't think many of you agree, but said, what is in the future? Because it takes 15 years and $2 billion to develop a new drug. What is down the road? And we looked at some of our own tumor samples, and in the literature, we discovered that in many of the cases when these taking away the brakes, the immune system go after the tumor, and when they win, of course, you don't see the tumor again. But when they lose, what was spectacular was you see this tumor like a soccer ball, and then you have a layer of cells called macrophages, and then thousands of layers of killer T cells. In other words, it's almost as if this layer of macrophages is indeed the wall that separates the Greek soldiers from getting into the Helen of Troy. And that brings me to what we are working on right now. What is the Trojan horse? What will bring these soldiers back in and then win the battle? And so together with Jan Berger at MD Anderson Hospital, we tracked many, many chronic lymphocytic leukemia patients. Chronic lymphocytic leukemia is a very interesting disease. It's actually chronic lymphocytic lymphoma because the tumors are actually outside, not in the blood. But the drug ibrutinib does is it releases that homing of the leukemia cell into the lymph nodes. When that drug succeeds, the cancer cells go back into the blood and basically die in the blood like a normal cell. So we hypothesized there was a Trojan horse that brought these leukemia cells in the lymph node. And so we isolated that Trojan horse, and we turned the table around and say, can we use that now to throw in and help those T cells who couldn't get into a tumor. And that's what we're working on.
Because I know that there's so much promise for immunotherapy, but heretofore it's been primarily with blood cancers. So what are the big challenges, TAC, when it comes to solid tumors? Because I know we've talked before about the tumor's microenvironment or the cancer's microenvironment. Is it because it's having a hard time penetrating sort of this wall that seems to be surrounding the tumor tissue?
Yeah, I think that our favorite cell is this cell called macrophages. Macrophages they are pretty normal cells. They're called monocytes in the blood. But when they go into Rome, as we did then in Rome you do what the Romans do. And the monocytes become a Roman. But if they go into a tumor, they will become a tumor macrophage. And they go into the lung, it become a lung macrophage. So if you remember when your kids were growing up, there was this story about this click, and the animal becomes something else. So those guys carry dozens of green lights and red lights. And when they are surrounding the tumor and preventing the immune cells from getting in, they have the red lights on. And what we need to do is to switch them to the green light to allow the immune cells to go in.
Are you all optimistic that immunotherapy will be as effective on solid tumor cancers as it's shown to be in blood cancers? And what are some of the biggest challenges?
So I am. I mean, we already see melanoma and lung cancer as being approved indications along with kidney cancer for these immune therapies. We know that others are very amenable, including head and neck cancer. And I think we hope that other very common diseases like very well differentiated breast cancers or colorectal cancer can be figured out in terms of immune targeting. I think that we're now also understanding that there are subgroups of people with those diseases who are very amenable to immunotherapy, and that brings up the importance of clinical trials, exploring the activity of these medicines in diseases that we otherwise might not think they would have activity in. There are some diseases, to continue a little bit with what TAC was saying, where there's a really hostile environment around some of these solid tumors. Pancreatic cancer, a disease that's very important to all of us, is one where the tumor is surrounded by a very hostile group of other cells, some fibrotic scar tissue and cells that actively keep immune cells out. And so we are working through this actually convergence program that you mentioned for a way around that.
Isn't epigenetics going to play an important role? That's always a little bit hard for me to explain, so Dr. June, why don't you do the honors?
So,
And am I right, by the way? I may not be right about the role of epigenetics.
So I think you're right. I mean, we'll have, it's going to be literally one of these, it takes a village. So cancer is complex. We shouldn't underestimate that. Some tumors have a much more toxic or poisonous microenvironment. They have this wall around that TAC-MAC was describing, comprised of cells such as macrophages and so on, which prevent the immune system from getting in and killing the tumor. And so we now know cancer is, in general, it's a genetic disease, meaning you have broken genes. Maybe 10 or 20% of human cancer is caused by viruses. But in the end, those work through genes to make the cells go haywire and become cancer. Over the last 10 years or so, there's been a whole nother level of the understanding of the complexity of the cells, and that's this epigenome that you're talking about. And it turns out some cancers are almost entirely caused by the epigenetics. So they really have low levels of mutations, but the way that those genes are controlled has been hijacked. So both targeting the hijackers as well as the actual genes that are broken will be ways to make new therapies. And
How does it get through, sort of like the candy shell, if you will, that surrounds the tumor?
yeah, so one way is actually to break down that shell And then basically it's an attraction just like a magnetic north and south poles. They'll come in and do their job. There are about 300 different kinds of cancer. So each one, it turns out, has different barriers that we need to study. So we need to learn from each other so that it will get to a point where all cancers have solutions. But at this point, I think what we do have is enough fundamental understanding to put together approaches that will lead to major advances, where in the past we had no idea where to go, really.
Hack and Jim, how has collaboration, we've heard so much of that discussed by Vice President Biden, by Sean Parker, by, Stand Up to Cancer. How has that changed your ability to move science forward faster in terms of sharing information and working with different institutions and scientists?
I think it's really accelerated the pace of advancement in the areas for a number of reasons. One is that there's a relatively small community of us, and we know each other. In the past, a lot of us have collaborated in groups of one or two or three in different institutions where people have different areas of expertise, different interests, different strengths, but by bringing together teams of people in larger numbers from larger numbers of institutions with a common goal, it really has been speeding things up, both in my experience with the DREAM teams and now in the early days of the Parker Institute. The goals are very similar, and that's to get people to find some tasks and say, okay, everybody, let's get going on that.
What about you, Ted?
I think it's essential, and in a lot of times we would like to work with each other, but we neither have the time or completely, want to trust the other person. But then you at Stand Up to Cancer, you come in with a check and made an offer that we cannot refuse, and it's also in Rome. No, that's not in Rome. And we do it, and it's spectacular. The results that, as you said, two drugs been approved because of this kind of collaborations. I'd like to see more of it.
Let's talk a little bit in the time we have left about secondary cancers. This is an area of particular interest to me because, having done stories on children with glioblastoma or various childhood cancers and even adults who are treated for a particular cancer, I always worry about secondary diseases down the road because of, what has to be done to their bodies. Often wreaks havoc with their systems. Is the possibility of secondary cancer is that decreased or seems to be decreased with immunotherapy because it's not sort of the scorched body approach that chemo is?
Yeah, I think it may be a little bit too early to tell for sure, but on basic principle, I think you're right. The prior classical approaches to cancer treatment based upon chemotherapies, which can cause the DNA to be changed and mutated, and radiation, which can do the same thing, are concerning because they can cause other cancers down the road. We know that patients treated with immunotherapy largely feel well. We don't know yet whether any of the inflammation that they had during the treatment might be related to problems in the future. They seem to be doing well now, but it's a good reason to continue to follow people really carefully.
If I could add something, though, I think that what Jedd was touching on is that these drugs, the radiation that cause the secondary cancers, are doing so because they're causing mutations. And we know now through a lot of work from a lot of people that it's the products of mutations that the immunotherapies are attacking, particularly with checkpoint blockade, that the T cells are going after. And so I think that there's no data yet, but there's talk about actually maybe taking advantage of these mutations induced by some of these chemotherapies to make more targets in the cancer and, in fact, treat the secondary mutations in the tumor or the secondary tumors that might come up later.
You mean like a combo therapy,
Yes.
chemo and immunotherapy in concert?
Right. Exactly.
Go ahead, Ted.
So I guess we all recognize that cancer is driven by evolution. Basically, you capture one of them and they found a way to escape, and you capture them back with a different method, and they found another way to escape. And you capture them again and again. Our patient would go after 4, 5, 6 different treatments, and they still escape. So they become serial escape convicts. So in fact, I think we should looking forward, be thinking of what are the traits of the serial escape convicts, and target those instead. And I think the field is getting close. A new pathway that is doing that is in this called the CDK2 cyclin E pathway. They are upregulating all the serial escape convicts. So I guess the thing is we have to be one step ahead of them. I think they are now 10 steps ahead of us, but we will get there.
Do you think one day that immunotherapy will completely replace things like chemotherapy and radiation, and those things will look terribly antiquated and almost inhumane in terms of treatment?
I think in some diseases, they already are close to doing that. So melanoma is the example where I can tell you we used to only have really chemotherapy to treat people with melanoma, and now I can probably estimate less than the number of fingers on my left hand, the number of people I've treated with chemotherapy in the past year or two. But that's not going to be the case for every kind of cancer. There is going to be the need for combinatorial approaches where some
I
cancer...
like that, combinatorial. That's a new one.
I don't think I made that up right now, but, but I think that sort of approach where you have something that directly affects the proliferating cancer cell, whether it's a chemotherapy or a targeted therapy, which interferes with sort of the circuitry that's wrong inside the cancer cell, that is going to be required for immunotherapy to achieve its full potential in some kinds of cancer.
You agree, Carl? And what are you most excited about as you look ahead and think about the potential of this approach?
There's a number of things that excite me. I mean, surgery, 2 of us up here recently have had surgery for cancer. So surgery is still going to be used, to cut out cancers. That's a fundamental thing. Now, surgeons came out of the field of barbers, people who cut hair. So when you look at medical history, I think what will happen is we're going to go to more and more precise therapies. So surgery is a precision targeted therapy. You cut out where the cancer is. Things like chemotherapy, where the whole body is treated, we're going to be moving away from that. And where I'm hoping we're going to go is, we're getting better and better technology where we'll diagnose cancers earlier and earlier. So unfortunately, many cancers we can't diagnose until they've already spread, as in your husband's case. And so we will have assays to detect these cancers sooner, and I think we'll actually get to the point where patients are vaccinated. Patients who are at risk for cancer will be treated before they ever have the cancer. So our goal that way is to have that happen. A number of cancers are familial. We inherit risk for them. And I think the technologies are going to get to the point where we treat in advance. So those require then technologies of better improved diagnosis, more precise therapies, and that's how it will be easier on our bodies.
Could I add something?
Yes, absolutely.
One of the things that I'm sure everybody here is familiar with is the notion of genetically targeted drugs that hit the driving mutation that causes certain kinds of cancer. So that's been the main goal of the war on cancer in the U.S. To sequence the genome and identify the lesions that cause the cancer, develop drugs that kill it, because that ought to be the perfect way, except it really hasn't been all that successful because the tumor cells keep getting mutations, and so you target one and there's another one and it's a mess. But what I think is going to be very exciting when we start putting these 2 things together, where you use the targeted mutation that can kill 99%, hopefully, or more of a tumor cell, but by itself, that other 1% will come back and it'll be resistant to the drug. But if you combine immunotherapy, you might be able to take it all out and have the immune system then go after all the other mutations that are in the cell and get really curative things. So you could go into somebody's sequence and say, well, you've got a EGF receptor mutation and we can target that, so let's target that and then give immunotherapy with it. And I think that it hasn't happened yet in any big degree, but I think that's going to be an exciting thing when it starts happening.
I mean, it's also exciting when you think about it. I wonder, since this concept, the notion of immunotherapy has been around for about 100 years, what took the scientific community so long to really embrace this notion?
I think we were really carefully studying what the molecular mechanisms were that were underlying what turns the immune system on and off, how cancer cells can take advantage of some of those mechanisms. There were some observations made in the late 1800s about connections between infections in cancer and life expectancy from the cancer that led to the hypothesis that if the immune system could be stimulated, that people might actually live longer after a cancer diagnosis. But we didn't know the science. It really took many decades of careful study, understanding what the immune system signals were, to turn it on and turn it off. And then we could develop targeted treatments within the immune system and develop engineered cells, such as the ones that Carl's are using, that use those same signals to make sure that the cancer cell is properly targeted and the immune cell that's going after it is energized to the greatest possible degree.
And was our knowledge of genomics and basic biology and some of the things that we've learned recently in cancer research, did that help accelerate sort of this notion of immunotherapy because it gave you much more knowledge about cell behavior and what certain cancer cells secrete? Not really?
I wish that we could say that it had, but it turned out there was such a chasm between the two fields that there was very little communication.
Is that
between
starting
the
to be
people.
broached?
Yes, that's starting to be broached now, but there was two areas of cancer research, and the immunotherapists, I must say, we were looked at as snake oil salesmen and
Yeah.
not really doing anything.
Well, you showed them.
I hope so. But no, the exciting thing is they're coming together now for the first time.
But we have one advantage, is that we have spent the last 30 years trying to understand how to shut off the immune system for the autoimmune disease situations. Now we just have to turn the coin around and say, well, what I'm going to shut off now, I'm going to boost. And simply that's in a very simple term. And we have a lot of those experience.
Two quick questions in closing. You know, it seems to me that, you know, talking about the future, Carl, as you were talking about kind of preventing cancer, is there a way that people here can make sure their immune systems are operating at full capacity? Because we all have sort of the potential for cancer in our bodies, right? It's a series of sort of genetic mess-ups where I guess the final straw is the P53 tumor suppressor gene doesn't work. I have no idea what I'm talking about, but I sound smart, don't I? But so how do we make sure that our natural immune systems are really healthy and able to quash potential cancer-causing genes, or cells? Does that make any sense, by the way?
So,
I didn't hear
each
a
of
yes.
us, I think, I mean, you're right about P53. That's a gene that is mutated in about half of all cancers. And our immune system, when it's properly tuned, can detect that and eliminate it at an early stage. So that's what we call immunosurveillance, finding a cancer, and in fact, you never know you had it. That we now can document, sometimes fortuitously, where someone may have had a chest x-ray, there's a spot, and then, or a mole that then goes away. So the immune system can work to reject a tumor without any interventions, but how can you maintain a high-functioning immune system is an area of active research. We do know that things such as exercise can help boost immune function, and but some people have immune systems that are better than others, and so how we can raise those is a big area of research.
And finally, just because Pope Francis is all about reaching out to the underserved, I'm wondering, how can the medical community do a better job of making these novel treatments accessible to everyone? Obviously, when I went down to MD Anderson to do a story, you know, it was very moving to see 2 women with melanoma being treated with immunotherapy, but it made me wonder about people in rural areas, people in inner cities, people who don't have means to be aware of these really exciting treatments, but also have them available to to those who are less educated or less informed and less fortunate? What do you, is there anything that can be done? Obviously, clinical trials is one way, but does anyone else have any other ideas?
I think many of the larger cancer centers do have outreach programs to try to bring their, I know Anderson does, to have, hospitals in the communities. I think educating people, though, that are not necessarily going to see the news or about these things
Right.
is an issue, and I think it's.
Well, if we could stop covering Kim Kardashian and Donald Trump, maybe we could tell people about this.
That would help. That would help.
Yeah,
So I just want to say that for immunotherapy, it's very different from small molecules. A small molecule, chemists spend 20 years making a molecule, they file, and nobody can get that particular structure. But because of the patent laws that says nature cannot be protected, you and I can go out and make an immunotherapy drug. The Merck, the BMS, the Pfizer cannot stop us. And so there could be certain kind of situation where philanthropy can come in and say, I'm going to make this drug and I'm going to give it out almost for free. And I think that will really address the question that you are asking.
I would just say that we have an example of, this health disparity and how it's being overcome has been in Africa, where we've had epidemic of, for instance, HIV, and people just didn't live long enough to get cancer. And now we have outreach programs, foundations such as Bill Gates and so on have really changed, I think, the destiny of Africa, and installing the healthcare systems where things then can be used to implement care for cancer now, which will become an increasing issue as lifespans extend there now. So it's something we're all going to face.
Yeah, I couldn't agree more. I think it's very important that we get these medicines to people who need them. I think, most patients are amenable to the idea of immunotherapy. They need access to medicines when they're not affordable. And I think the important other topic is clinical trials, because that can often be a source of medicines for people who don't have them. We need to increase the dialogue, really around the country about the importance of considering a clinical trial.
I know that's a big goal of Vice President Biden's because, as you said, 3% of patients participate in clinical trials, and there's a lot of misunderstanding about placebos versus actual treatment, which really isn't the case.
Yes, and we have a lot of explaining too about things that were done wrong in the past, and what the potential benefits are of being in a clinical trial.
Well, gentlemen, thank you very much.
Go ahead,
Tad.
I just will make one before I shut up. HPV, the vaccine has done so much for reducing the instances of uterine cancer, head and neck cancer. It's a vaccine. You take it, you don't have to worry about it. And that's cheap, and that will be so much more effective in the long run. And this is just my point to say that that wouldn't make a difference whether you're rich or young, but you'll be protected.
Well, gentlemen, thank you very much for your insights and everything you do every day. Thank you.